Thank you for joining us. Today, I'm very excited to invite Dr. Vince Calhoun. So this is our third molecular connectivity online series. In case you are new here, so we are a molecular connectivity working group, which is a group of researchers from a diverse background, from biomedicine to health science, neuroscience, and physics. So we aim to validate advanced molecular imaging as a tool to study brain functions and to validate these methods. So we organize this online series in order to promote scientific discussions about research on molecular connectivity. And we plan to host this online series monthly for every third Friday of a month. You can subscribe to our website and newsletter to get the latest information. Today, I'm very pleased to introduce Dr. Vince Calhoun. He is the founding director of the Institutional Center for Translational Research in Neuroimaging and Data Science. He is the author of more than 1,000 full journal articles. His work includes the development of flexible methods to analyze neuroimaging data, neuroinformatics tools. And personally, I have used a lot of ICA-based methods to study PET data and fMRI data, and a lot of tools are based on like group ICA or other ICA-based methods are based on tools developed by Dr. Calhoun. Today, Dr. Calhoun will introduce their newly developed methods of NeuroMark PET toward a fully automated PET ICA pipeline. Once again, thank you for coming, and welcome, Dr. Vince Calhoun.
Great, thank you. It's a pleasure to be here. So let me just jump right in. So what I want to do is sort of motivate this pipeline a little bit and show some results that we've obtained from PET data. And then towards the end, I'll also talk about integrating it a little bit with fMRI data as well. So just kind of a brief intro, which many of you may already know. Oftentimes, the atlases we used are based on anatomic regions, and they're typically fixed, even if they're, we take an atlas, we register it to a subject, and then those are fixed, and you're either in or out. And those atlases are very useful, very powerful. However, they have some limitations. For example, they don't capture variation in those, ROIs among individuals. And in particular, if you're doing functional imaging, you might be averaging together voxels which are functionally distinct. So we know that functional boundaries don't correspond one-to-one with anatomic boundaries. And we also know that, if you do any brain network analyses, those networks can overlap with one another. And in PET data in particular, we can have different tissue types like white matter that will have, some overlap at the boundaries between gray matter. So ICA is one of the most widely used data-driven approaches in fMRI. And it's an approach that allows you to extract maximally independent sources, spatial sources, which can overlap with one another. It's been successfully used in PET as well, in dozens of studies at this point. However, one of the challenges is comparing across studies, because every study, is doing a fully data-driven approach, and you might have, different components in different studies. So just sort of a brief review. ICA is basically organizing the data in the context of PET like this, where we have a single PET scan that's been pre-processed and normalized into a standard space. And it's organized, the voxels in that single PET scan for one subject are organized along the row of this data matrix. And then we have multiple PET scans or multiple subjects in each row. And we're decomposing that with ICA into a set of spatially independent sources and a mixing matrix. And so a row of this matrix here is going to be a component map. And so that would be something, it'd look something like this if you threshold it. And then this mixing matrix contains loading parameters, which give us subject-specific information about the component. And to go into that a little bit more, those loading parameters also coming out from the mixing matrix, I like to describe it as they represent the relative degree to which each ICA component is expressed in a given subject's PET image. So for example, if this component is strongly present in a given subject, the loading parameter will be high. If it's not very present, it'll be near zero. And if it's negatively present, meaning it's subtractive, it would be a negative value. So that's the loading parameters. We can take those loading parameters and do statistical tests on those. So we can do, for example, if we have a 2-group study, we can organize the data like this, and then we can, for each component, test for differences in the loading parameter values between controls and patients. So that you could do with a 2-sample t-test, you can do regression, you can do any statistical model you would like on these parameters. Analogous to what we do with fMRI in the context of functional network connectivity, we can also look at the cross-correlation between the components here. And that gives us information about the degree to which these different networks or components are correlated with one another or linked to one another. And so here's an example in a 53-component case where we have component by component matrix. So I'm kind of overloading this FNC term, and for PET, we'll call it functional network covariation. So we're really looking at the covariate. If 2 different components have covary similarly between between one another, then that correlation value would be high here, and these are anti-correlated. And so you can see this modular structure, and I'll talk more about how we ordered this, but that's another source of information that we can get out of ICA. And this kind of thing, I think, is less common in the use of PET. And so just some examples of just a low model order ICA. These are from floor beta peer PET data, and you can see we get you get often things if your PET tracer is present in gray matter regions, you often get networks that look like this. Where you get bilateral, they look kind of like resting fMRI networks. We get default mode network. We might get a few lateralized components, for example. So a couple slides on pre-processing, and I'll touch on some of these data sets later on in the slides. But we're going to be looking at a couple different tracers, amyloid imaging, as well as FDG PET. And we're using this open NeuroPET pipeline, where essentially we take the PET data and we convert it into a BID structure. We do motion correction. We use FreeSurfer applied to the T1 to do co-registration and normalize into standard space with PET Surfer. And then I'll touch on scaling later, but for data-driven approaches, it's common to use like a standard scaling, like you might z-score your data, or you could also use standardized uptake value ratio, like you might in other PET studies. For example, scale each image relative to the average activity in the cerebellum, right? So then that's relative change to a given region in the brain. And here's an example of what you would get from a floor beta peer amyloid image. These are controls, are healthy individuals, and you can see that most of the uptake is coming from the white matter. We also have gray matter uptake. In the case of individuals with Alzheimer's, you start to see the gray matter regions show more uptake over time. Okay, so now let's look at blind ICA. And so again, reminding of this perspective here, our mixing equation is X equals A times S. So that's the equation. If we just take a second and talk about scaling, because that can be an issue that comes up in PET data, it's I think a really nice result here is if you just work through the math of, say here's our data matrix X, and suppose we want to scale each individual relative to their cerebellum, right? So that would be a single subject scaling parameter that would be applied to all of the voxels in that image. And so that's matrix B here, which we just multiply by X to apply that scaling. And if you then take X and you replace it with AS and kind of work through the math, in the end, the sort of take-home message is that the sources that we get are invariant to the scaling if you're just using a single scaling parameter for all of the voxels. The scaling is only impacting the loading parameters, and you can do that after ICA. So if you want to explore different types of calibration for PET, you can do that after the ICA. You don't have to rerun the ICA a bunch of times. So that I think is a really nice, gives us a nice way to do comparisons. We often get nuisance components that come out. So here's that dominant white matter that I showed you earlier comes out in a few components. Here's just an example of one of them. And of course, the component has values everywhere, and it can overlap some with the gray matter. So it gives you a way to essentially remove white matter from the analysis if you take this component and subtract it from the data, for example. Here's a picture of just an example of a number of gray matter components that came out. This was a 40 component decomposition of that Fluor Beta Peer data set that I mentioned earlier. And you can see we get, we cover pretty much, most of the gray matter. I'm not showing you the white matter components or the CSF components here. However, we can use those, as I mentioned. So if we start with this data set, we apply ICA, and then we subtract out the white matter and CSF components, and then reconstruct the data set, you get this here, which now, these are, again, controls. So even in the controls, you can start to see the distribution very clearly in the gray matter areas. And then you could take this image and just analyze it in some other approach if you wanted to. So you've kind of denoised, or it's sort of like a partial volume correction, ICA-based partial volume correction. If we just take the pre-processed PET data and do voxel-wise analysis on it, so there's no ICA here, and we look at a relationship to age, you get something like this if you ignore, white matter. It's obviously dominating, right? So you do want to probably deal with that. But if we look at just the gray matter regions that show increases with age in controls, you can see that you get, cortical areas. You get a little bit of probably default mode network here, for example. That's a voxel-wise analysis without ICA. If instead we look at the components and we categorize them into those that show decreases with age in their loading parameters and those that show increases with age, you can see consistent with the prior slide, we have, kind of a default mode network component, a lot of cortical components, but there's a little, there's a mixture of directionalities that come out that you can see much more clearly in this ICA decomposition, because we're now we're analyzing the data kind of at the network level as opposed to at the voxel level. All right, so now I'm going to pivot for a second and talk about NeuroMark, and I'll start with our fMRI framework, because that's where we've been using a lot of it. We've been using it a lot for fMRI, and so the basic idea is let's generate sort of a PET component, a set of component templates, and we do this by taking several large data sets, run blind ICA separately on those, and then look at the components that show consistency or that replicate across those data sets. And then we use those to build a template that contain components. And then once we have those, we use a variation of ICA, which maximizes both for independence as well as for similarity to the template. So that's a spatially constrained ICA approach. And so we've applied this to fMRI, and it's really useful, I think. First of all, once we have derived the template, we've got the components and we've grouped them, into sort of anatomic categories. It's really just ordering of the components. And then we can run the spatially constrained ICA. And I think one of the nice benefits, in addition to it gives you the same components or similar components that are adapted to a new data set, it also gives you them in the same order. So you don't have to, so here's an example of two separate analyses. And this is functional network connectivity and resting fMRI. And you can see very consistent, sort of main effect resting patterns in these two independent data sets. And then if we look at more subtle effects, in this case, we had a schizophrenia data set, we look at patient versus control differences because we get a matrix like this for every subject. And we can look at the group differences in those two data sets. And you can again see the patterns. They're a little noisier. These are small effects, but they're fairly consistent. We get, and they're highly correlated to one another. And so not only do you get out this, components that are matched, you also get out components in the same order. So you don't have to reorder your components. You don't have to select what's good, what's bad. And so the pipeline can be fully automated. You simply just give it the templates, give it the data, and it'll give these results out without any intervention, manual steps. We've compared this approach to, fixed ROI approaches. If we look at just a few standard ones like the AAL Atlas or the Brainatome Atlas, and we applied a classification algorithm, we found that the data-driven or sort of spatially constrained ICA NeuroMark approach outperformed the Atlas-based approaches. And we continue to develop this. So here's a newer template. The one I showed you before was 53 components derived from Human Connectome Project and Genomic Superstruct Project. Here we're using 100,000 subjects across many different studies. So the idea was a broadly defined template, try to make it as generalizable as possible. And we wanted to look across ICA model orders. So we ran all of these different and ICAs with all of these different number of components. And then we looked at identifying components that were replicable, and we didn't duplicate components. And the final atlas gave us 105 multi-scale components. And there's a picture of what they look like. And this is our FNC atlas that we get as a result from that. And I think the last example I'll give you is, again, we've got a bunch of stuff in this, and that's not really the point of the talk today. But I tend to prefer approaches that can be applied to look across studies as general as possible. But questions often come up is, do we want cohort-specific templates? I think those can be useful in some cases, although they have limitations. And so we did look at this. We've generated templates, for example, for infants, for developmental ages, for aging. And you can see they're actually highly similar to one another, with only a small set of components that diverge from one another. And you can apply the same approach to other modalities. So we can do this for gray matter data. We can do this for white matter fractional anisotropy data. We can do it for structural connectivity as well. So there's a bunch of stuff here. I'm not really going to cover all that, but just to kind of highlight that context. And within our GIF software, we also have tools for doing this in an automated way. And so, for example, we've created a BidZap that lets you access this pipeline. And you can implement a fully automated ICA approach, or you can just run it in the GIF software in MATLAB as well. Okay, now back to PET, which is, can we take this framework and apply it to PET data so that we can get consistent results across studies? And so we have, if we can derive a PET template like this, and then take a new data set, apply spatially constrained ICA, and then we would get out spatial maps, subject loading parameters. We can do statistical testing, and then we have a comparability to that to prior studies. So here are there's 3 columns here. The first 2 columns are they were just split into 2 because there were a lot of them. We have different components that fall within gray matter regions. And then this third column contained white matter CSF components that were not included in the template. Row 1 and row 2 are 2 independent fluor beta peer data sets that were separately analyzed. And then here you can see consistency if you look between them. Some of the colors, I think, are not exactly the same, but you can sort of hopefully see that they're quite similar to one another. And then here is the final template that we generated that included those gray matter regions. And so just to give you one example where we've evaluated this is we could take that template, which was generated with fluor beta peer scans, and apply it to another data set with a different tracer, in this case, fluor beta ben, which is also F18, fairly similar mechanism. And the question was how similar would it look in terms of this FNC compared to the fluor beta peer? And you can see it looks very, very similar across these, fairly similar tracers. And I'll give you some more comparisons to contrast with this in a second. And we can also we can use this template and we can look at, for example, Alzheimer's versus controls if we have data from those 2 groups. Here you can see the components shown around a connectogram, and then here is the FNC between them. And this one of the stronger connections here, which was showing essentially higher uptake in Alzheimer's, is this link between posterior hippocampus and middle temporal regions. I'm also showing you the results from 2 different scalings, where we have essentially a Z-score approach, and then here's that cerebellum SUVR approach. The overall gestalt is similar. There's a few extra links that show up, on either side. They're not identical. They're fairly similar. So the scaling does have an impact, and so that gives us, I think, a reason to maybe optimize over the scaling, for example, or use your preferred scaling, whatever that might be. And that's FNC, but we can also look at the loading parameters. And you can see here each bar here is a component, and these are 53 components. And I forgot to mention this result. Well, yeah, so we have 53 components here, but notice how we get a lot more uptake in Alzheimer's versus controls, as we might expect, right? And so a lot of those are quite significant. Okay, and so the last section I wanted to talk about was going across modalities and across tracers. And I'll start with one, which is fMRI informed PET ICA. And so here we're taking resting fMRI networks that we identified from that first NeuroMark paper that I mentioned, and applying those to PET data. And so you can see here, I've just shown you 3 example components on the top. After we do that optimization, you can see they're not identical, right? They're optimized for the PET data, but they do match. And the ordering here of the FNC is also matched. And you can see this is our resting fMRI connectivity pattern, and this is our PET covariation pattern. And so this gives us, I think, some really interesting ways to start to compare across modalities. Here's some broader examples. This is all using fMRI networks and to inform the PET decomposition. So it gives us a reference to compare against. Of course, we don't have to do this. We could use, for example, fluor beta peer as our reference, or we could look at all these combinations. And so you can see some of the differences here. So here's the fMRI. here's that floor beta peer data set I mentioned. Here's the floor beta bend data set. You can see how similar those look to one another. Here's a PIB PET result. And here's an FDG PET result, which looks more similar, at least in this central modularity, I think, to fMRI. It doesn't have these strong, and I should have showed you the domain labels here, but this block is cognitive control and default mode network right here. And this is subcortical. That's where most of the differences are showing up. So that is, I think, all I wanted to cover in this talk. We, of course, have this GIF software that we've made available, so you can feel free to download and play with it. You can, generate your own templates if you want to try the spatially constrained ICA, or use the templates that we've already provided. We've got all of the fMRI templates up at this point. We're still working on the PET templates, but those will be coming. And so with that, I want to just say thanks to all the folks at Trends who did the majority of the work here, as well as collaborators in the Open Neuro PET project. And here's just a subset of many folks that were on the team. Cyrus, of course, is with me at MRN. This is Melanie Gans and Cyril Purnett and Martin Norgard, who developed the PET Surfer BIDS PET framework. So with that, I will go ahead and stop. Thank you very much.
Thank you very much, Dr. Calhoun. It was a very interesting and inspiring talk. So now the floor, I think, is open for questions, for discussion. You can either raise your hand, I think, in the participant list, or just write in the chat, and we are going to gather. So. Okay, so Arianna.
Yeah, so great talk. I have a difficult question. So you just mentioned at some point that one could try to optimize the scaling. And here, I think it's a gigantic topic, like what scaling you should use, because like with FDG, you have many different options that are all considered acceptable. So like, what could be a criteria to say this is the scaling that I want to go for?
Yeah, no, that's a great question. I think it depends on your question at hand. So for example, if you're, I guess a relatively easy one, quote unquote, is if you're interested in classification, right? So you could that's your criteria, and you can run classification and sweep all of the possible scaling parameters in that sense. Of course, there are many other ways to do that. And of course, just because it classifies well doesn't mean it's neurobiologically meaningful. So there's all those sorts of questions you've got to keep in mind as well. But I think it's a framework that you could really use in many different contexts.
So if there's no question for now, I have a quick question with respect to these constraints that you can apply using constraint ICA. So how strong are these priors? How stringent are they? And how can you potentially modulate the strength with which you rely on the prior versus instead rely more on the data?
Yeah. So that comes up a lot, and we're using sort of equally weighting independence in the priors. That's a parameter you could play with if you wanted to. We've looked at this in the context of fMRI, and it is in between. So it's not data driven completely. It's not model based. So it's not fixed. It does adapt quite a lot to individual subjects. And so we've played with cases where we don't have a source in there and it's in the prior, right? And then you do it and you get these kind of random looking things. So it's not forcing it to look exactly like the template. So it is adaptive, but it's in between. It's a trade-off, right, between the two. So yeah.
Thank you.
No free lunch, I guess.
Can I ask a question? Hey, hello. Thank you very wonderful talk. So I have the question is that if we use PET, right, usually we assume that it measures something maybe different from both. Or if we use different tracers, and then usually we want to have something new or novel, right? And then so can we assume that like the template we generated from both, how well it can be applied to this PET data? And yeah,
I think that's a great question. I mean, as you may have seen from the examples I showed, the sort of covariation patterns differ quite a lot depending on which tracer, and they differ quite a lot from fMRI, right? So there are a lot of differences there. What you're doing, if you can think about just going back to Tommaso's question, you can think about a spatially constrained approach as sort of like a adaptive ROI, right? So you've kind of, so it's not unlike I take a fixed atlas, right? And I look at correlation in those same regions in fMRI versus PET, right? The only difference is that we've got more flexibility here. We've adapted those ROIs to the PET data or vice versa. And also, I would say, I don't think it necessarily always makes sense to use fMRI as your constraint. You might want to use, just want to look at like, that's why we're developing PET specific atlases, tracer specific atlases, right? Because there may be some particular patterns of covariation that are unique to those data sets.
But maybe we need a large sample size to do that, right? So in terms of sample size, what is your opinion on?
Yeah, what we've been playing with, I mean, you can do this in, I mean, you can do ICA, of course, on 20 or 30, subjects, and get reasonable looking networks out for low model orders, right? You can't do the higher model order stuff in that case. For the results I showed here, we were dealing with several hundred subjects. So it's not that many. And again, we're trying, the more the better, typically, I would say. And if you get more data sets and you can split them into independent replicable, replication studies, that strengthens, gives you more confidence that you've got something that will generalize. So.
Thank you.
Mm-hmm.
So now we have two questions in the chat. So Shengyao Wang says, great talk. Is this method suitable for a single subject analysis?
So in the PET case, we have, if you have a standard PET scan, you might have only one, after you've combined, your different scans together. And so, yes, you can apply it to that, but what you would get out are single parameters. So you would get out loading parameter for that subject. You're basically just fitting the maps to that data set. You wouldn't get optimized spatial maps in that sense, because there isn't really much to optimize over. You could potentially do more if you had dynamic PET, or if you had multiple PET scans per subject, and then it should work in that case. And you would get more information, spatial information, than you would from just a single scan.
And Mohammad Ali Yavanrey says, many thanks for the great talk. How interpretable would be the functional components obtained from the PET data compared to the extracted values from the structural templates? Also, how different or generalizable would be the findings from one site to another?
Okay, I want to make sure I understand. So the first part's talking about when you say structural templates, are you talking about gray matter templates? Is that correct?
Yes.
Okay. Yeah. So if you use you mean like anatomic templates, like, so if you have like a fixed ROI, right? No, that's a great question. I think it would be good to compare those side by side. I didn't do that in this talk, other than mentioning the fMRI comparison we did. So yeah, and you know, I think there's two things. I think that you will get. I believe it will be more sensitive because of the fact that you have this overlap that you're not getting with the fixed ROIs. And then the other thing is you don't get out the spatial. So the component maps will vary. They can adapt to unique data set. You don't get that information at all in a fixed ROI based approach. But yes, it would be good to compare, how do these covariation patterns look, right? Do they look similar? You wouldn't expect them to be completely different, right? So and then what are the pros and cons, et cetera, right? So everything's a trade-off. And then in terms of generalizability, I mean, at least in the context of NeuroMark PET, we have looked across data sets. And so it is very consistent across data sets. And that and so that is kind of one of the goals of this type of a pipeline, is to try to get more generalizable results. And we could certainly quantify that on more data sets should we have. But you saw how similar the maps looked in the two different floor beta peer data sets. So those were independent sets of subjects.
Okay, so we have a question from Nakul Raval. Do you see this as a replacement of traditional ROI-based analysis or a supplementary method to this, or more focused at using different data sets together?
I mean, everything's got a pro and a con. I think it's a complement. So the ROI-based analysis gives you the ability to pick exactly where you want to look, right? Whereas with these templates, you're adapting to the data. So the data is telling you where you should look. And so sometimes you want to do the former. Sometimes you may want to, do the latter. And so I think they're complementary.
Hello, can I ask another question? More in general question. So for example, if we have two groups of subjects, we want to compare the two. Usually we perform ICA on this together, right? Because we do not want to bias. Like we only perform ICA on this healthy subjects. Then if we apply it will be a little bit biased. So, but in terms of this NeuroMark approach, you have all the data to generate this template. But that template is actually all based on healthy subject, I assume. And if you apply the methods to like even my own data, we have two groups, then will that be introduced some bias about only healthy, those templates are only based on healthy subjects?
I think the range of variation that we typically see is within that which can be captured by the ICA optimization. If you remember we've done a lot of these kinds of comparisons on the fMRI side, right? And so those group differences I mentioned that we highlighted were clearly obvious in a template that was designed and developed from healthy controls, and it adapted, and we can see not only differences in F and C that were highly sensitive and also gave us predictive accuracy. Like we could classify quite well, 85 to 90% with those results. It also gives us subject-specific spatial maps as well that showed differences between controls and patients. So yeah, I mean, I think that's the, but I think the other end of the coin is that 100,000 template that I mentioned for fMRI. There we're including everybody, and that at least we had access to. It's not everybody, but it's a set of data that from open repositories that we're able to put together to try to capture a broader swath of variation. And so that was exactly kind of one of the motivating points that we had was let's try to generalize this to not only just this slice of controls, but just everybody different ages, different groups, etc.
Thank you.
Yeah, let me get that paper for you. I will paste that. Someone had asked for that 100,000 one. I just put it in the chat. So.
May I ask a question?
Mm-hmm.
Related to what Xin also asked, is like, so even for amyloid PET, so PB, florbetaben, florbetaben, you also based the template on healthy controls or Alzheimer's disease patients?
The one that I showed you was based on controls.
Yeah, because I was a bit wondering, like, what's the biological meaning of that? Because like in healthy controls, yeah, I mean, you can have some healthy control, cognitively healthy controls are amyloid positive, but some of them will be amyloid negative, so really not have much uptake in the brain. So did you check for that? And, or like, do you see any meaning, say any specific reason to still look at these based on controls instead of patients?
Well, I think it should be able to adapt across that range. We didn't do a comparison yet of positive and negative, amyloid. We certainly could do that and do more. I think we were just looking at, we started out with controls, we looked at age variations, and we also look at this extreme case of controls versus patients. We're just getting started with this, so that, we haven't done all of those comparisons yet. But yeah, my opinion is that it should be able to adapt to those differences within subjects. So the control template is, in a sense, close enough of a starting point for the algorithm to adapt to an individual that has variations across their spatial maps. But that's something we can test. So.
Thanks.
Mm-hmm.
Can I add another question, a provocative one? So last month we had a talk by Chris that was arguing about how good it is to use PCA, right? This month you come and you tell us, no, guys, ICA is very cool. So could you two guys just start fighting right now and just let us know what's the best approach we should use?
Oh, PCA is horrible. No, I'm just kidding. It's not. It's not. No. They're different. They're just different approaches. I guess, yeah. Okay, you want us to fight? Okay. Well, no, I would say I tend to agree that you can look at the data through different lenses. I like ICA because we're looking at higher order statistics, and we're incorporating not just variance, but we're also incorporating higher orders to separate out components. So it's not just trying to push all the variance into a low dimensional, basis set, so to speak. That being said, I think there are, useful ways to use the PCA solution as well. And there are lots of other options that you could decomposition approaches that you could apply there. So, so I don't know. I'm not that controversial, I guess, on that point. But
Actually, I'm also not controversial. I have no problem. I think maybe I'd rather fight with Vince against deep learners a little bit more, because I still believe in rather flat architectures and decomposition. Also, by the way, I really like bringing hyperparameters and all of this to the fore, because people sort of forget about this, and it's also, I think, a great didactic tool that you can play. You make sparsity constraints stronger or weaker. I would, and maybe this is more philosophy. I wonder what you would say about people who now present foundational models of deep learning. I tend to be a little bit suspicious about deep learning or much deeper learning architectures. So I don't see much of a difference. So ICA imposes higher order independence. That's fine. I have no problem with that. We're just doing slightly different things, but I think we're both much more similar than some of the deeper architectures. I don't know whether you comment on that.
Yeah, no, I think we're still early in the deep learning phase, even though it's been around it's been in the neuroimaging space for about 10 years, but most in the past 5 years, I would say. And so it's still early. So foundational models, I think, are, not yet. One of the reasons I say that is because there's this missing piece of understanding what the model has done. We've been actually working a lot in that space as well. And there actually are a lot of different cool things you can do to visualize, but we're using like these networks that we can get from the flat architectures, like you said, to initialize deep models. So we have a reference point. But I just think right now there's so much happening in so many different, every paper has a different, like the problem I mentioned of within ICA, comparing across 2 different studies is exponentially worse in the deep learning space, because you can't compare one study with the next because it has a different architecture. They all are different. We don't have yet a full handle on how to decide what's useful and what's not, and how to connect it well to the neurobiology. And so I think it's still early. I think there's a lot of potential. I actually see a lot of potential there, but I think there's a lot of overclaiming that's happening right now. So
yeah, it seems.
I think that it's ironic that the algorithm is deep learning, but the papers don't go very deep into what they're studying, right? It's really just a lot of it are just algorithm papers that have neuroimaging tacked on.
Right. Yeah, it's
my cynical view, but yeah.
I always find it funny, and I definitely think that the proof is in the pudding, and ultimately it might be great, but it's also, I find it funny when people, and I had this on a study session, I obviously can't say who it was and all that, but when somebody was very excited about a foundational model and almost seemed to suggest that nobody else had done large-scale meta-analytic sort of, flat architecture decompositions on large data sets before, and that struck me as very odd.
It's the new toy on the block.
Yeah, I mean, it's kind of people forget about history here, right? I mean, as if nobody had done some stuff before, Anyway, so.
Well, the ICA algorithm we use a lot is Infomax, which is a single layer neural network model, and you can extend that to multiple layers and start to look at nonlinearity gradually, things like that, right? So there are things you can do there. I think there's a lot of cool potential out there. But the first thing I usually do with a new paper is, oh, it's really cool. It sounds like there's something going on, and I skim it, and I see no images, and then I'm like, okay, I don't believe it, because I need to see visually. I need to visualize, because this is because of working with many of my students, seeing that first pass, and then we dig into it, and more than half the time it's like, oh, it doesn't make any sense at all. You have to tweak this, you have to tweak that, you have to figure out how to make it work. And if you don't show that you've done that due diligence, then I'm not convinced that model is useful at all. So.
Yeah, also I mean that would be my last sort of editorializing. I find it interesting because to me often these architectures fall from the sky rather than, I mean, you mentioned a way of building them up, right? Which makes sense. You introduce layers of complication, but I always see they're kind of God-given, and I can't appreciate why, so.
I mean, I think there's 2 ways to do it. I think you can build from what you have, or you can say, here's something that I want to do and I can't do it with this, and so I'm going to create a model that will let me get at that. But you still need to compare it with some standard approaches. Is that enough fighting, Ariana?
Yes, it's very unsatisfactory fighting, but I'm glad that you agree. We should invite an AI expert next, and then
Well, at the next meeting, I'll drink Vince under the table. Yeah, yeah.
There we go.
Definitely. Yeah.
Maybe to go back to the topic of the presentation, one question I had for you is like, so I know you worked in the past about kind of merging the information from different modalities to have a sort of joint ICA, right?
Mm-hmm.
So in this, if you think about PET, what you think it makes sense to merge with what, and like, you know, to get more, what would be useful to do, like?
Oh, that's a whole nother talk. So yeah, data fusion with PET data is very rich. There's a lot that you can do there. And I think you can usefully combine it with fMRI, with structural MRI, with diffusion, with all the MRI modalities, with genomic data, with all sorts of things. So there's lots of ways to do that. So that's a rich topic, I think.
Yeah, hello. Thank you for the great talk. Can I also ask something regarding the presentation?
Yeah.
Thank you. So the template you presented is that you had like 58 common networks, right? And so if we want to create our own template from our data set, what do you think should be the criteria that we can say, okay, this is a good template to use for the later?
So the floor beta peer template I created was not, so the 53 component one was an fMRI template, which I used at the end. But earlier in the slides, we actually did develop a floor beta peer template. And so we had, I think it was a 40 component or 50 component decomposition we used. And we essentially had two stages where we ran on roughly equal groups, two different blind ICAs. And then we identified components that replicated across those analyses. And then we categorized them into, white matter, CSF, and gray matter, and some artifact, type components. And then of the gray matter ones, we then ordered them, based on their locations. So that was kind of the process we followed for it, which I know you're familiar with as well. And I think that extra piece of, if you have enough data to do two different analyses on independent subjects and look at replicability, that's an additional, I think, useful thing to do, because it gives you more confidence that it'll generalize.
Okay, yeah, thank you.
But you start with whatever data you have, so.
Are there any more questions for Dr. Calhoun?
Yeah, I think if there's no questions, I think we are about time. Thank you again, Dr. Calhoun, for his wonderful talk and the discussions. Yeah, don't forget
Thanks all. Yeah.
Follow. We have this symposium organized, and also if you are interested in follow our newsletter to get more information. Thank you, everyone.
Thank you.